Hereby, I open this academic ceremony in which Mr. Català will defend his academic thesis, Understanding the Complexity of the Corneal Endothelium for Regenerative Medicine. And I welcome you, of course, and wish you lots of luck together with your paronyms. And I welcome, of course, your supervisors, Professor Nuits, Dr. de la Pointe, and Dr. Dickman. I welcome the members of the opposition, who you will meet later on. And of course, welcome to family and friends in the room, on site and online. But before we start the opposition, the floor is yours for a short presentation about your work.
Dear Prorector, members of the Corona, family and friends, thank you all for coming here. And in the next 15 minutes, I will be presenting a short summary of my research in the past 4 years. But first of all, explain a bit what the cornea is. The cornea is the clear window to the world. As every other tissue in the human body, it has a highly structured shape that fits its function, and it allows the light to enter the retina, and thus it allows us to see. My work reported in this thesis focuses on the most inner part of the cornea, the corneal endothelium. This is a monolayer of corneal endothelial cells that reside in contact of the Descemet's membrane in the cornea, and these are actively transporting water from the stroma into the interior part of the eye, maintaining the cornea slightly dehydrated and transparent. But corneal endothelial cells remain in a quiescent state. It means that they lack proliferation capacity, meaning that external stressors such as aging, Fuchs dystrophy, surgical complications, and infections can cause an accelerated cell loss. This accelerated cell loss can compromise the barrier function of corneal endothelial cells, leading to corneal opacity, as you can see in the image on the right. And can you imagine how it would be to live with corneal opacity? It would be something similar that you can see in this slide. Nowadays, the state of the art for treating corneal endothelial disease is selectively transplanting the damaged corneal endothelium with that of a donor. But there's only one donor cornea for every 70 patients in need, leaving 12.7 million people awaiting corneal transplant. The aim of this thesis is to study different platforms to tackle this problematic, providing treatment to those in need. And we would like to do that by maximizing donor tissue use, but also by developing novel regenerative medicine therapies. Nowadays, 70% of the corneal transplantations are lamellar, meaning that only the most inner part of the cornea, the corneal endothelium, gets transplanted. And the process looks something that you can see here in the right, where basically only a small roll of cells is transplanted in front of the eye. In chapter 3, we studied if it would be possible to already transport from the cornea bank into the clinics a pre-rolled corneal endothelium graft already in a ready-to-use insertion cannula, instead of sending the full cornea. These devices look something similar to this. Well, basically they look like this, where the corneal endothelial graft is preloaded and soaked in media. We performed this study by transporting the corneal endothelial grafts that were pre-stripped in the ETB cornea bank in North Holland, and transported them from until the Maastricht University Medical Center. Using these devices is important because it would allow us to maximize donor tissue use. It means that the rest of the cornea could be used for other processes, but also it would reduce the iatrogenic damage of this tissue at the moment of surgery, meaning that the pre-stripping of the cornea, peeling the corneal endothelial graft, would be only performed by highly skilled and trained people in the cornea bank. Our results show that there were no differences in between the conventionally pre-stripped cornea, which is basically the full cornea, towards the preloaded cornea in this graph. Next, we understood that for developing novel treatments, it is crucial to gain knowledge on corneal biology. And traditionally, the corneal layers have been studied as bulk entities. We were lacking tools to analyze for their full heterogeneity until the arisal of single-cell RNA sequencing technologies. So in chapter 4, we performed single-cell RNA sequencing technologies on human healthy corneas. And to give an example of why these techniques are important, I would like to make this analogy. Well, the cornea is heterogeneous tissue, and this is similar to a smoothie, where you have different fruits. And let's say that you want to gain information from an orange or from a strawberry. But instead of taking that orange or that strawberry, you mix it with a pineapple, you mix it with a banana, you blend it all, and that's what you're tasting. It's going to be really difficult to understand what flavors are coming from that specific orange, or for instance, from that specific cell. That's what happens when you're doing bulk RNA sequencing. Single cell RNA sequencing would allow us to get that orange or that strawberry, or that cell in this case, and get all the information that we want to know. And by using this technique, we would like to identify all the cell types in the human cornea. We would also like to identify layer-specific markers to be able to differentiate in between corneal populations, and also use this technique as a benchmark for developing future regenerative medicine approaches of the human cornea. So again, in chapter 4, we developed a protocol for dissecting healthy human corneas, dissociating them to single cells, and perform single cell RNA sequencing. Our results show that the cornea is a highly heterogeneous tissue. We were able to identify 9 different cell populations in the most outer part of the cornea, the corneal epithelium. We were able to identify 4 different cell types in the middle layer of the cornea stroma, and 2 different cell types in the most inner part of the cornea, the corneal endothelium. We also leveraged the knowledge gained by this data to be able to identify novel markers that were previously not described. These markers were specific for identifying highly proliferative cells in the periphery of the corneal epithelium, but not in the central part of the cornea. On the next chapter, we focused more specifically in a regenerative medicine approach. This was a cell therapy, and nowadays it's possible to isolate corneal endothelial cells from donors, expand them in vitro into numbers that would allow us to treat multiple patients from one single donor. And this is a reality nowadays in a clinical trial in Japan. But there are some limitations preventing the adoption of this technique worldwide, such as this technique is only successful when isolating cells from very young donors. And this is especially difficult because getting young donors, it's not so often that you have them. There's a lack of clear quality markers to identify what cells could be used for patients, and there are alterations over extended culture time. For this reason, in chapter 5, we apply the same technique that we established in the previous chapter, single cell RNA sequencing. We isolated corneal endothelial cells from different donors, culture them, and sequence them at different time points to identify what alterations were appearing when we were expanding them in vitro. Our data set is shown in here. Every single dot that's represented in this map represents the genetic signature of one single cell. And we were able to identify greater quality cells in cluster C1 or C0, and lesser quality cells in C2 and C5. We also show that there was a transition from the cells of good quality towards the lesser quality cells. We also again leverage the information that we gained performing this analysis to be able to identify novel markers that would allow us the identification of the cell populations that we were aiming at using for therapy, and other cell populations that were not desirable for therapy because they were losing expression of functional markers. Finally, in chapter 6, we studied if the differences in the animal kingdom could aid the development of novel regenerative medicine approaches. And interestingly, rabbits possess a proliferative corneal endothelial cell layer, meaning that these cells are highly proliferative upon injury. This is different towards bigger mammals, such as humans or sheep, where the cells are arrested and do not proliferate. We were really curious and trying to understand what's the mechanism driving this proliferation in corneal endothelial cells from rabbits. And for this reason, we isolated corneal endothelial samples from rabbits, humans, and sheep. We performed bulk RNA sequencing analysis and also transcriptomic pathway analysis. Out of this comparison, we were able to identify 52 different pathways when comparing human against rabbit corneal endothelial cells and sheep against rabbit corneal endothelial cells, meaning that probably these 52 pathways could play an important role in triggering cell proliferation. And most importantly, we focused in 2 pathways, notch signaling pathway and TGF-beta signaling pathway. Finally, in chapter 2, we perform a literature review on the state-of-the-art therapy, meaning corneal transplantation, but also we give a perspective on novel therapeutic approaches for regenerative medicine of the cornea. So in conclusion, we discovered previously unknown cell markers within the healthy human cornea. We identified undesired cell types appearing in the in vitro cultivation of corneal endothelial cells. We also identified new ways to identify high quality corneal endothelial cells that could be used for therapy. And we also identified some of the genetic drivers that trigger proliferation in corneal endothelial cells in other animals. We also showed that grafts, corneal endothelial grafts, can be preloaded and transported from the cornea bank into the clinics safely. That would allow us to maximize tissue use. And I would like to finish my presentation by showing this slide. This is a slide to portray the importance of science and to portray the importance of the role that we are playing here. In 1953, the application of hot air was used for treating corneal endothelial disease. Nowadays, a cell therapy has become a reality, but there's much more path that we should continue working on to make these therapies available to everybody. And I would like to thank you all for your attention, for being here. And I would like to give back the word to the prorector.
Thank you very much, Mr. Català, for this nice presentation. You kept certainly in time, gives us a little bit more time for the opposition. And the opposition will be opened by Professor Sluimer, who's also the chair of the assessment committee. She's professor of cardiovascular pathophysiology in the University of Maastricht. And she's always also the secretary today of this committee, for which I'm very grateful. Professor Sluimer.
Thank you, Prorector. Dear candidate, first I would like to congratulate you with the content of the thesis. It was a pleasure to read it, although my field is, as you know, quite different, vascular biology. I was very much intrigued and I learned a lot about the cornea. So very, very well done for making me understand that. Also impressive papers, awards, grants. So I think well done on that. So let's see, of course, how we are going today. And I would like to also congratulate the team with this big team effort. And I really also enjoyed the fact that the Merlin and the Eye Center here are joining forces, which you can see that definitely this brings forward the field much further than the hot air for sure did. About some seriousness as well. First of all, though, I would like to start with the proposition. And I'm wondering if you or one of your paronyms would like to read proposition number 7.
EMT is the monster hiding under the bed for scientists.
Thank you very much. So for the audience, EMT is cells losing their identity, going from one cell type into another. And so I have to disagree with this proposition, and for the reason that I think actually this is great. As a scientist, I think it's marvelous that there's something to do. It keeps me busy during the day, makes me sleep like a baby with an angel over my bed. So I was wondering if you can maybe tell me about your statement and why did you come to this proposition?
Dear highly esteemed opponent, thank you first of all for your kind words. Well, this proposition is pretty much field related, and there's some fear in ophthalmology field of NMT and also of fibrosis. It's not specific to other fields, but having cells losing their functionality or losing their tight junctions, it means that these cells gain a proliferative capacity and gain loss of function. They are not going to be behaving as they should. Of course, in science, you have other examples where actually an EMT is favorable. And this happens, for example, in embryological development when there's the definitive endothelial migration. That's a positive aspect of an NMT. This is a field related. It's more specific to ophthalmology. I should have maybe put it more clear in there.
Yeah, that's very clear. So you're talking about a field related clinical perspective, and it's not a good thing. I can understand that. So staying a little bit in this area, and as well about the single cell sequencing. So when I'm doing a single cell sequencing, I hear more and more from colleagues who are not doing it that they're a bit skeptical. You know, it was all great in the beginning, but everybody's doing descriptions and that's it, right? So I think we should really make more opportunity of these data sets, and especially the one that are so unique like yours with all the human data. And we need to move towards causality studies or moving it into a therapy or something like that. So I would like to discuss that with you, and if you have ideas how to take your work further. Specifically though, you describe in chapter 5 You have the different subsets on page 80 if you would like to look 180 if you would like to look it up, of the cultured corneal endothelial cells. If I understood correctly, CT and C4 are undergoing EMT, and C0 and C1 you think are usable for transplantation. So with the knowledge you generated, how could you use this to ensure more cells for transplantation?
Well, basically, we show that we can select different markers. We did this only at the immunofluorescence level, but the information that we gain in here would also allow us to sort the cells that are positive or that we think that could be used for therapy. That's the first thing. Another thing that we are showing is that there are some gene signatures or gene hallmarks that are popping up, and this probably can guide the improvement of primary culture protocols.
And how do you envision this guidance?
There were some pathways popping up, for example, the ROGTPase pathway. Nowadays, there's discussion if we should really use ROC inhibitors for primary expansion of corneal endothelial cells. In my view, and showing the results that we obtain in here, yes, we should use them. There's a lot of ROGTPase pathways that are appearing in the more senescence or more NMT clusters. So using that could be a solution. Something that we also saw is that there was an increase in TGF-beta expression. There's also some protocols from Japan that they use TGF-beta inhibitors, specifically the SB43. So maybe looking into other small molecules. For example, a ELK2 inhibitor would be an example from top of my head. But just, yeah, these are just examples I could give.
Yeah, they're already very interesting. And have you already talking about pathways and genes, and have you also looked at transcription factors that can drive those pathways or maybe prevent EMT from happening?
Yes, we didn't do that much in depth. This can be done, of course, with the same data set, but we just decided to keep it as it is. There's other techniques that could be especially interesting in here based on single cell RNA sequencing. For example, is PRDD-seq. These are novel techniques that are appearing now. And these techniques allow us to perform lineage tracing or cell tracking. So these techniques are especially powerful. We needed something to benchmark all these techniques against. That's what we did in here.
Yes.
But now we open the door for future research.
For more. Well, I'm glad to hear you have such good vision for the future. Also, staying in this chapter, I noticed that there is a lot of endothelial cells in there, but also in the chapter 4, the in vivo study. And I was wondering how comparable are your cultured endothelial cells to your in vivo cells? Yes,
that's indeed something that we did in chapter 5. I don't recall exactly the figure, but we did 2 types of analysis, which basically one was to portray where these clusters would position in the same data set of the cultured cells. They were positioning very close to clusters C0 and C1, but we also did a predictive study of what clusters would be these native corneal endothelial cells be representing. The results were that they were representing, they were falling in cluster 1, cluster 0, and cluster 6, which was the good quality corneal endothelial cells.
So now I need to get the numbers straight. But so there was an EMT cluster also in the in vivo situation.
A tiny bit. There was a 0.3% of the cells that were falling into that cluster.
0.3%.
That's correct. It can happen. It's something that can also happen in native tissue. It can be happening if, for example, there's a scarring in that corneal cell, or even during the isolation of these cells that we perform, we can trigger this type of reactions.
And what is the evidence, the in vivo evidence, or based on in vivo tissues, that there is EMT going on in the in vivo situation?
Well, that's indeed a very good question. There are some examples that when the corneal tissue gets injured, it's unable to proliferate. And I'm not fully sure if it's NMT or fibrosis, but it's happening also. I don't know if when there's transplantation, for example, of different devices, I know that clinicians are very scared of this fibrosis. So they all fear fibrosis. That's something that I've been hearing in this field. So it's happening not only in the corneal endothelial layer, but also in other corneal layers.
Yeah, so hence the monster. Yeah, I guess in the only way to really prove it, which is impossible in humans, is doing reporter studies or fate tracing, lineage tracing, other. And is there any evidence in your animal models, any of the animal models with a lineage tracing that is pointing towards EMT?
We even do a lineage tracing analysis, but that's something that, of course, can be done, and it's very likely that will appear.
And others in the field, have they used a mouse? And
I haven't seen research like that.
Okay.
No.
Well, it sounds anyway an interesting phenotype that, or like a bad monster that you want to prevent. Pro-Rector, do I have some more time? Or maybe a short question. In chapter 4, I was looking actually for a UMAP to show the sources of your cells, so the different donors. And I didn't really find it, whereas you do mention it in chapter 5. And I was wondering, how did you confirm that you're not looking at batch effects rather than cell type effects?
Every time that there's every time that we sequence the cells, we perform a donor correction. This is part of the study that was done by our collaborators at the Hoover Institute. So I'm not familiar what type of R script they used, but there's a donor correction.
And have you made the UMAP with the sources? And
yes, it just locates everywhere. I think that we also show in the supplementary data in chapter 5.
Yes, yeah, there you do show it indeed. Yeah. Okay, well, thank you very much. And I'm happy with the discussion, and I would like to give the word back to the Pro-Rector.
Thank you very much, Professor Slimer, for your position. Now we come to the following opponent, as Professor Scottman. She is professor in the Faculty of Medicine and Health Technology in the Tampere University, and that is in Finland. And thank you, Professor Scottman, for coming all the way from Finland. The University of Maastricht is always glad to have guests from abroad.
Thank you, Pro-Rector, dear candidate. It was truly my pleasure to read your thesis, a very interesting topic close to my heart. So first of all, of course, I want to congratulate you and your supervisors that you have reached to this point that we are here today to examine your work. So while reading your summary of all the work you have been conducting, from transporting the tissue to the transcriptomics of full thickness cornea, transcriptomics of cultured primary cells, also including additional animals there. So I was really impressed about the amount of work you have been conducting during these years. There are many things I would love to discuss with you, but let's start. As you know, I'm a stem cell person and I love markers, cell markers. So let's discuss first a bit about the markers for corneal endothelial cells. As you summarized also in your thesis, Professor Kinoshita has his own panel of markers, and then they have updated that with this kind of erasure exclusion kind of panels as well. So based on your own scientific findings in your work, so how would you revise or make that panel better?
Dear highly esteemed opponent, thank you very much for your kind words. Kinoshita's panel, I have to say that it's really well developed. It's really well established. We can, of course, make it better. It would be an example using some of the markers that we are showing. Chingulin-1, it's a membrane-associated protein. This was highly expressed in better quality corneal endothelial cells, but also excluding markers. I believe Kinoshita is using an array of 4 to 5 exclusion markers. From our findings, not all of them were represented at the transcriptomic level, but of course they need to be assessed at the protein level, which is the most important. But also we are showing some of the exclusion markers that could be added in there. The example is THVS2, and another marker that we could add in that array.
Yeah, thank you. So overall, how do you think about how good is the negative marker as compared to positive markers? So
that's indeed a difficult question to answer. The problem of corneal endothelial cells is that most of the times you don't have a yes or no. We are trying to find those golden markers to do that. It's very difficult to find them. Not even Kinoshita markers are yes or no markers. What we have seen though is that there's high differences in between CD166, for example, in good quality corneal endothelial cells, and those of lesser quality. The same is happening with Chingolin 1, and that's why I was so positive about this marker. Like there's a huge difference, even though it's tiny bit expressed in the lesser quality cells. The same comes from THBS2. And these markers, they're more yes or no. We don't see them at all on good quality cells, but we do see them at lesser quality cells.
Yeah, okay, thanks. Then also your summary, you were discussing about the potential limitations of single-cell RNA sequencing. And you mentioned there that we can miss up, for example, some rare populations, like you weren't able to see the quiescent limbal markers, for instance. So what do you think? Are we missing maybe some rare endothelial cell populations as well? Could there be stem cells, at least in young patients?
That could be indeed a scenario that we actually contemplated. Out of the 19,000 cells that we sequenced, we only sequenced 800 corneal endothelial cells. And this is due to the difficulty of generating this single cell suspension. Corneal endothelial cells are extremely sensitive and they don't like to be on single cells. So it can be that we are indeed losing some cells. To be able to increase the significance of the study, we should, of course, add more donors.
Okay, thanks. And then regarding my background is in animal biotechnology, so I was really keen to learn about your study where you were including the animal species for transcriptomics. So, but I was puzzled and wondering, why did you select a rabbit as a model for proliferating endothelial cells? Why not, for example, mouse, where we have a lot of genome data available?
We actually tried to perform this study in mice. The problem is that getting a pure isolation of corneal endothelial cells from mice, it's almost impossible. So we tried the pilling approach, we tried enzymatic dissociation, we sent the samples to sequence, but they were always coming back with stroma contamination. There was expression of lumican, keratocan, so it was impossible to get a clean isolation of corneal endothelial cells from mice.
Okay, and then quickly about that study. So why did you select bulk RNA sequencing there, not the single cell one?
Well, this was a proof of concept study. We didn't know where we were putting ourselves, first of all. We just were curious of, are we going to be able to see what differences are in those animals? So we just went for something that we had more expertise or we were more comfortable with. Even though we thought it was going to be straightforward, it wasn't. There's a lot of limitations when you are doing this type of studies, for especially bringing together all the data sets. In the end, I'm very happy with the results that we get, but of course, if we would like to continue in future, there's other research that can be done with this interspecies comparison.
Okay, thanks. Then one more stem cell related question. So as you know, in my lab, we are differentiating corneal endothelial cells from human pluripotent stem cells. So based on your results, how would you help us to improve the efficacy of our differentiation method?
It's actually a very difficult question to answer. I'm not fully familiar with the reagents that you're using in your protocol, but what we have seen is, for example, using ROCK inhibitors, it's important for the primary expanded cells. I'm not so sure if that's going to be the case for stem cell derived corneal endothelial cells. What can help though is the markers that we show. These are another validation step to show that actually the differentiation protocols are generating cells that we believe are corneal endothelial cells. And I would encourage you to use the markers that we show in this research.
Okay, thank you. Do I still have, Prorector, time for one question?
Yes, certainly,
Okay.
certainly.
So maybe kind of last question from my side. What would you do differently if you would start all over?
General question of PhD, what would I be doing differently? There are actually many things that I would do differently. Here I'm showing part of the work that we've done, but there's many research that has been ongoing that didn't make into this booklet. An example is trying to make a corneal endothelial cell carrier. We were never successful. I think it's because we selected a technique. It was too difficult for the purpose to get a transparent corneal. We were using electrospinning technique. It was almost impossible to get transparent carrier. So that's something I would be doing differently. There's also some research that I would like to do differently, knowing the findings that I report in my thesis, and that's especially in the interspecies comparison chapter. What we did is isolate RNA from healthy tissue and just perform an interspecies comparison. I would do something different though, now that I have seen the findings, is not, in order not to lose as much information as you can lose, I would get the tissue from the healthy animal and also a tissue of a healthy animal where we do a scratch test. Then isolate the samples from those animals and perform a differential expression analysis, and then compare those pathways and see what actually is happening when there's an injury in the cornea.
Okay, thank you. Okay, thank you, dear candidate. I think I'm very satisfied with your answers, so I return back to the project rector. Thank you.
Thank you, Professor Scottman, for the opposition. Then we come to the following opponent, who's also a member of the assessment committee. That's Professor Bachmann. He's professor in the Department of Ophthalmology in the University Clinic of Cologne, and that is in Germany. And welcome, Professor Bachmann, and thank you for coming all the way from Cologne, although it is online, but more than welcome, and the floor is yours.
Yeah, dear Pro-Rector, thank you for the invitation to this committee. Dear candidate, it was really a pleasure to read your very comprehensive thesis and also to hear your excellent presentation just now. And I want to congratulate you and your supervisors for this excellent thesis. And I really love to read these different chapters, which I think really well complement each other one to the other. And I also want to ask some questions with regard to chapter 5, which I think is one of the most impressive work you present within your thesis. And there I also read that you used cold cultured corneas, which you received from Temple. So a very basic question would be, so why did you decide to use cold cultured corneas? Because in Europe, we generally use warm cultured corneas. And why you use these corneas, which came all the way from the United States?
Dear highly esteemed opponent, thank you very much for your kind words. We used cold storage corneas because we were using a protocol that was established in Singapore. In that protocol, they use cold storage corneas. So we wanted to use it as much close as the clinics it would be. They're using this protocol also for a current clinical trial. So we wanted to replicate to the most the setting that they have in there. Of course, if we would like to make this research translatable to Europe, it has to be repeated with organ culture corneas.
Is there anything now on how your results could be transferred to the warm cultured conditions? Are there any implications from now in research so far?
Sorry, I didn't hear the question properly.
So are there any information how your results from cold cultured from corneas from under cold cultured conditions, how your results could be used for our warm cultured corneas? Or are there any publications which show that there are some diverse effects of cold culture on your main results?
Actually, we have performed also in our lab. So I can speak from firsthand. I can also give some reference, which I will do later. In our own hands, we also try to culture organ culture corneas. We don't see any difference in between cold storage corneas and organ culture corneas. There's also research from Mohit Parekh that he shows that he's able to isolate these corneas by using the same exact protocol and culture them into numbers that fit the numbers that we're getting.
So within this chapter, you showed that different clusters occur with different properties, and you showed that there is a certain cluster, which was cluster 3, which has an increased proliferation capacity. And you suggested that this higher proliferation capacity is due to the transition to fibrotic cells, could you also exclude that there are also maybe some corneal endothelial cells with a higher proliferation capacity within this cluster, which might also be suitable for therapy?
It's indeed highly proliferative corneal endothelial cells. But what we show in cluster 3 is that part of that cluster, so within that cluster, we identified 2 subclusters, which one was proliferative corneal endothelial cells that we believe are still useful for therapy, and a small cluster within that cluster, it has markers of endothelial to mesenchymal transition. So actually it's both. We have cells that are proliferating that show corneal endothelial cell markers, but also cells that are proliferating that increase over time that show these markers of an endothelial to mesenchymal transition. So actually, to reply your question, I think it's both. It's especially important to portray this to show that actually in the long run, if we keep culturing these cells, it might be that the cells that underwent the endothelial to mesenchymal transition overgrow the corneal endothelial cells. Because something else that we saw also is that the proliferative cells decrease over time. So it can be that if these cells that underwent an endothelial to mesenchymal transition keep proliferating, if we would passage further than passage 2, that's what we did in this research chapter, they would overgrow the better quality corneal endothelial cells. And that's something that we don't want.
A further question would be about chapter 6. There you could demonstrate that 2 pathways are more active in non-proliferative human or sheep corneal endothelial cells when you compare them to the rabbit corneal endothelial cells. When you look back to your work from chapter 5, could you also find any change during the culturing conditions and during the different passages you performed with these cells with regard to these 2 pathways? Did they change in activity? Could you make any conclusions on that?
Yes, not for notch signaling pathway, but for TGF-beta signaling pathway. Chapter 6 can be linked to chapter 5, where basically we saw that the cells that were undergoing the endothelial to mesenchymal transition have an increase in TGF-beta expression. So that could be something that could link these chapters. That's also something that's ongoing. For example, in the protocols for corneal endothelial cell expansion from Japan, they use TGF-beta inhibitors. So this basically links the pathway that we found in chapter 6 and the pathway that we found also in chapter 5.
Okay. I also have a question to chapter 3, where you compare different transport conditions, the pre-stripped and only DMAC grafts and the preloaded DMAC grafts. And you couldn't find any difference in baseline donor characteristics. Was this achieved by accident, or did you perform any kind of randomization between the 2 groups?
We didn't perform a proper randomization. We were just getting some tissue from the cornea bank. These are the findings that we found. It's not a proper randomization, no, but we tried to treat the tissue on the same way. We didn't find any type of differences in that way.
Okay. And during the preloading condition, the endothelial cells could grow on the other side of the desmoid membrane. This has already been described. So could you also find something in your specimens you investigated?
Still. Sorry, could you repeat the question? I didn't hear it properly.
It was shown before that under these preloading conditions, the endothelial cells can grow or migrate on the other side of the desmoid membrane. Was this something you could find in your specimens you investigated? Can you hear me?
No, we did. Yes, no, we didn't investigate that aspect.
Okay. Yes, so I think I'm finished with my question, and I want to thank you for all your answers and discussions, and I want to return to the prorector.
Thank you, Professor Bachmann, for your opposition. And then we go to the following opponent, who's also online, and he's also a member of the assessment committee, and that's Professor Seitz. He's professor and chairman of the Department of Ophthalmology in the Saarland University Medical Center. That's also in Germany. And thank you, Professor Seitz, for coming all the way, albeit online, but more than welcome. And the floor is yours.
Like to welcome for an excellent piece of work. I'm a clinician, I'm a corneal surgeon, so I could not understand everything, but what I understood was very well done and very well put together. It was in 2005 that Henry Edelhauser thought he had discovered stem cells of the human corneal endothelium. What do you think about that? Are there stem cells of the human cornea?
Ongoing discussion. We didn't find them in our data set, but again, as I explained to Professor Scottman, it can be that we might be losing some important cell populations in there. I would expect that if there's indeed this stem cell population of corneal endothelial cells, which might be indeed possible, I would expect them to find a few cells. So then we would need to sequence a high amount of corneal endothelial cells to find them. Regarding our data set, we didn't find them.
You have heard about this model of decimal stripping only. So what do you think results in the clarity of the cornea? Is it just migration or proliferation?
Sorry, I don't know if I properly the question. Is the audio, it's not amazing. You're asking about DSO and what do I feel about the...
Is it just migration or is it also proliferation that leads to the final clearing of the cornea?
Again, an ongoing discussion in the field. I think it might be a bit of both, actually. By using ROCK inhibitors, we might be triggering proliferation of corneal endothelial cells, but as well triggering migration. That's likely because I think that DSO, correct me if I'm wrong, it's only successful in very early stage patients and relatively young. So maybe these cells still possess small proliferative capacity and high migration capacity, plus the use of these ROCK inhibitors might be of help. So I think it's a bit of both.
You showed the role of the hot air in the beginning, and patients typically see worse when they wake up in the morning. So what is the reason for that, and how can hot air help? Can you explain that?
The reason is because you sleep with the eyes closed, so there's a greater inhibition of water inside of the stroma. By using hot air, you dehydrate slightly the cornea, so hence you make it a bit more transparent. There's some hospitals that still recommend in their guidelines this technique, and you still find it in some websites that they actually recommend for really mild cases to blow dry your cornea in the morning. So I think it actually works. It dries a bit the cornea, it makes it more transparent, but not in the long run.
Yes, excellent answer. Other question: if you do DMEK, decimal membrane endothelial keratoplasty, and in contrast, you use the penetrating keratoplasty. Can you tell me how over 5 or 10 years the endothelial cell deterioration of the graft happens?
Not very familiar with penetrating keratoplasty. I'm more of a scientist, but my colleague that previously defended the thesis showed today some results that he found a graft failure rate of 85% after 1 year, which decreased to, correct me if I'm wrong again, to 70% after a few years after that. I'm not familiar though with the full penetrating keratoplasty, so I cannot answer this question. And
how about the deterioration of the endothelial cells after DMEK? Do you know that? You know, in the timeline, what happens the first months, the years to come?
I think I'm a bit familiar, and most of the fast deterioration happens in the first months, and then it kind of stabilizes. But again, I'm not a clinician, so I don't have full knowledge to provide a well-built-up answer.
Yeah, I think it goes down the first months and then it stays more stable. Can you talk a little bit about the advantages and the disadvantages of the preloaded endothelial cell grafts? What are the pros?
Yes. The pros, I would say that it would allow us to maximize donor tissue use. I know that, for example, bulk transplantations are not very much performed, but we could still use these corneas for that transplantation where the inner part is for DMEK transplantation. It also would allow us to send from countries that have a proper organ bank facility to other countries that wouldn't have a proper organ bank facility. So we would be also providing tissue to other countries that actually need them. On the downsides, I think the very downside that I see it's with the trypan blue staining. I've received many grafts after transplantation. Well, many grafts, no, it's 2 grafts after transportation that they lost the trypan blue staining. If you have to restain that graft inside of the insertion cannula, wash it, and then insert into the patient, there's a lot of shear stress. So it might be that we are actually losing cells. So it has some advantages, of course, but it also has its disadvantages.
And how long do you think you can keep it in the preloaded device? So how many days would you see to be acceptable?
I think we performed 2 to 3 days. Study from Stefano Ferrari and colleagues by 2020. They performed 3 to 4 days, and they transported the grafts from Italy to Liverpool. So it's also a cross-country study. So that might be 4 days, I would say. That's what has been reported. Maybe it's more, but it has to be studied properly.
Are you aware of some long-term outcome studies comparing the preloaded shipped tissue in contrast to the directly prepared tissue directly before surgery, as I would do?
I'm not aware. I'm not aware of these clinical outcomes, no.
Yeah, because I'm skeptical, because I'm preparing that, by my own hand in the operating room, and then I know what I'm transplanting, because you never know what type of technician is doing that. Do you know a little bit about preventing the upside down implantation of the DMEK donor?
So I really couldn't tell how to prevent it.
Okay. And my last question would be, we have donor tissue shortage. You mentioned that many times in your work. So from your perspective now, what would be the aspects of getting more transplantations done in Europe, but also in the developing countries?
Well, my perspective is that in Europe, we are in a pretty good position where we have enough tissue. Most of the corneal tissue shortage is in Africa, in Asia, in Japan. It has a lot of tissue shortage. The main advantage of this is that, for example, Netherlands or Italy, we are net exporters of corneal endothelial tissue. So something that I think needs to be encouraged is having a communal attitude, and all this leftover tissue that it's not being used, exported to other countries that actually need it. I think it's especially important to maybe implement techniques for other countries that don't have a stable organ banking facility to receive these corneal endothelial cells or corneal endothelial grafts to transplant them. And the preloaded is one example of them. So I think we need to have a more communal attitude.
I still have a question. I'm working a little bit on and we try to expand stem cells of the epithelium. So if you compare that, expanding the endothelial cells and the epithelial stem cells, which is more difficult and why?
It's indeed I would question. I can only speak from the side of having tried to expand corneal endothelial cells. These are extremely difficult to culture. It's not only the culturing, but it's also the isolation. The isolation, it's a very tedious process where you have to carefully peel corneal endothelium not to get any type of contamination from the stroma. Dissociate them with a mix of collagenases. You don't want them to become single cells, you want them to become small clusters so then you can expand them. And the proliferation rate is very slow. So you have to nurture them, take care of them over time. So it's a pretty tedious technique and it's difficult to achieve. I'm not familiar with the culture of epithelial cells, but thinking from the point that there are stem cells and they proliferate fast, I think that would be somewhat easier to at least make them proliferate. Might be still difficult to isolate them, having a pure population, but I think that the proliferation aspect of these cells is better than the proliferation aspect of corneal endothelial cells.
Thank you very much for your good answers. I think it's 1651. Thank you very much.
Thank you very much, Professor Seitz, for your opposition. And the following opponent is also a member of the assessment committee, and that's Dr. Carlier. And she's an associate professor of the Faculty of Health, Medicine and Life Sciences in the University of Maastricht. Dr. Carlier.
Thank you, Prorector. Dear candidate, I would also like to congratulate you with a very nice thesis. I very much enjoyed reading it. I also learned a lot about the cornea, which is not my specialty. But it was very interesting. I thought also you did a very nice presentation and I very much like the artistic cover. Now, of course, I also have some questions related to the thesis, as is always the case with a nice thesis. And considering my background, I would like to go a bit more into the computational aspects of chapter 4 and 5. Specifically, so you, of course, based on the data, you calculate these clusters, which you then use to find markers. And so I'm just curious whether you could explain a bit more what was, let's say, the distance measure or the algorithm that you use to define these clusters.
Dear esteemed opponent, thank you very much for your kind words. The pipeline that we used, well, I have to say that this part of the analysis was not done by myself, it's done by our collaborators at the Hobrecht Institute. The pipeline that they applied is the Seurat pipeline, and they applied the differential gene expression within that Seurat pipeline. Out of this, we could select different clusters. We were playing a lot, and we had a lot of discussions in what cluster depth we should have. And basically playing in these aspects, we were asking ourselves, what's the biological question that we want to get there? And if we went too much in depth, we couldn't even tell what differences would the cells have. And if it was too much in bulk, we didn't even see biologically relevant questions. So to answer this question, it's the Seurat pipeline, differential expression analysis, and we played a bit with the sequencing depth. No, not sequencing depth, apologies, with the clustering depth.
Yeah, thank you. Yeah, and then also you did, of course, later also a gene set enrichment analysis. And yeah, you do that, of course, on the differentially expressed genes, but how do you define which genes are differentially expressed or which cutoff did you use?
Yes, we use a p-value of 0.01 on the differentially expressed genes. And that's what the cutoff that we got. We were getting lists of 500 genes that then we just ran into this pipeline.
And did you also explore different cutoff levels? Like how much did then the list expand or reduce?
No, we stick to the p-value of 0.01.
Yeah, okay. Now, moving then on to, let's say, a bit more the markers you identified, or let's say in the data set. Now, one of the things that I'm personally also interested in is, let's say, cell material interactions. And I can also imagine that the transparency of the cornea is tightly linked to, let's say, the organization of the extracellular matrix. So I was wondering whether you also saw some extracellular matrix related markers in your data set.
We do see them. Actually, in the native cornea, chapter 4, we show different populations of, in this case, corneal keratocytes that were more active in the production of collagen ECM. We also see some populations of the corneal endothelial cells that are more active in the production of, I don't recall exactly what collagen, but we thought that this could have a special importance in the maintenance of the cement membrane homeostasis. So we do see that the cells are producing ECM molecules.
And do you think that maybe making use of this ECM production, or that you could use actually this to improve the culture of your primary cells?
Yes, it's to answer this question, it might be indeed important for the culture. I think it has another application, though, that it's for transplantation. If we are able to generate a monolayer of cells that produces their own Descemet's membrane, it would be amazing if you could transplant that. There's some research ongoing on that aspect. The problem is that the cells do not generate enough of the ECM. What happens is when you try to lift this monolayer of cells, they kind of shrunk all together. So it's physically impossible transplanting them.
Yeah. Okay. Thank you. Let's see. What are the questions? Yeah. Yeah. Another question that I had is, yeah. So you do, of course, the RNA-seq, and yeah, then you do the whole data analysis. And as is known with, say, these kind of high throughput techniques, you often, of course, need to also validate what you find. So I'm wondering, yeah, what kind of validation experiments did you already do, or what kind of experiments do you feel could still complement the work that you already did?
Yeah, so I will take the example of the protein expression for the differential expression genes. We basically found that there were some differentially expressed genes in different clusters that allowed us to isolate or differentiate from higher quality cells to those of lesser quality cells. We did a first proof of concept with proteomics. Well, not fully proteomics, it was immunofluorescent staining. We only did that with 2 donors. Of course, it should be tried with more donors, and especially I think that it would be very important to show that we can assess them at the flow cytometry level and actually quantify our findings, either flow cytometry or Western blot. So if there's follow-up research that I would like to do on that aspect, is perform flow cytometry or Western blotting to actually quantify the expression of these markers.
Thank you. Yeah, to kind of follow up a bit on your question, so you already mentioned that you use different donors, and there's of course also donor variability. And what actually was interesting or very nice to see in your thesis is that all of the analysis that you did contains actually male and female data. But as far as I could find, you don't analyze the potential sex differences that are there, although you have the data to do it. So I was wondering whether you expect any sex differences or not. Are there actually, is it known that there exists sex differences in corneal pathologies?
I think it's a very valuable point. It was not the main scope of our research. What we wanted to portray is to do a first proof of concept analysis in human healthy corneas, not taking into account the female or male isolation part. I think it might have an impact on the corneal endothelial cell primary expansion, but again, it was not the scope of our research. It was just a proof of concept done for the first time. Wanted to show that actually we can track the changes that arise from the primary expansion, but indeed that's something that we should look into in the future.
Yeah. And then maybe for my final question, could one of the paronyms read proposition number 8 for me, please?
With the future increase in the therapeutic repertoire, it is critical for clinicians to select the best treatment option for each patient.
Yeah, thank you. Yeah, so could you maybe comment on this proposition, and also do you see some potential options for digital twins as a solution for your proposition?
Yes, I put this sentence because the default procedure up until now for corneal transplantation or for treating corneal endothelial disease was just DMEC or lamellar transplantation. I think that now we are starting to have some cellular therapies, some pharmacological modulation approaches, so even the start of some possible gene modulation approaches. I think that it's especially important now for the clinicians to actually decide what's best depending on what type of
You may finish. You may also say enough is enough.
I can surely finish. I think that it really depends on the patient that you have. And in regards on what disease stage they are, you should select a suitable therapeutic approach. So then we're actually providing treatment to the people in need and reducing a bit the burden of one donor every 70 patients in need.
Thank you.
Thank you, Dr. Carlier, for your opposition. And Mr. Candidate, Mr. Català, the time appointed for the defense of your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and of your defense. And I request that you and your company, on site and online, await the results of our deliberation in this room and our return. And then we will say what we decided. I suspend the meeting.
Good job. And what was really bad, it was the connection. Yeah, I couldn't even understand the question. Was an accident. Yeah. Yeah.
You did try to avoid the first question to say it's a good question. I think you meant to say it's a good question, and then you stopped and you you said what I very nice one.
Done.
Like super professional. You didn't invent anything.
And it's a thesis research from Cambridge. So I don't know all the outcomes.
It was good.
I think it was good. I like it. I enjoyed it.
Presentation was spotless.
Yeah. There was one question only that I hesitated from the first. It was the beginning.
First was the most.
But I have to say that two of the questions, I saw a presentation of a guy last week. And one of the questions he asked. So I already knew what question said.
That was nice.
No, it's from the hospital. They were my name.
Nice. Good one, yeah. Professional. It was professional.
Okay, guys. The glasses.
Yeah.
Yeah, I will do the diploma. I have a diploma.
Para pôr na parede.
Una prueba para él.
Yeah. Diploma. In the beginning, it's very tense. I think I will shake in the beginning when you see your family. And,
For me, what happened is that I was checking the time, and after the second minute, I 40 minutes.
yeah. It was fast. Yeah. It's a lot of time. It's a lot of time.
I think the doctor, the clinician, you couldn't see the time. And that's why it was.
Yeah, could be.
It's normally, well, like for some, you were not allowed to.
Yeah. You're probably.
Done. That's fine. You couldn't be a football player. You're a doctor now. See,
It's a trade-off, right?
it's a trade-off. It's not bad. But spotless. Very professional. Really enjoyed it. Because sometimes like people try to avoid, I don't know, so they try to, but in the end you said no.
People try to script their information to make something out of it.
Yeah.
Polite way to say it.
Yeah, we have to do the scientific I think it's now. That's a new thing, right?
I feel like since I was here.
Okay. I think standing.
Yeah, I think you should start.
Yeah. Still will take some time. Wait, I thought I had, the itchy that you make you cough. No, if this starts, will be awful.
Oh, nice.
It's well, you have tissues here. And then you give to a person. Yeah, thank you.
I reopen this academic session. Mr. Català, the degree committee here present has assessed the quality of your thesis and the way you defended it. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. And Professor Knights is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Pere Català Quilis, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. And the laudatio will be given by Mo Dickman. Mo?
Thank you, Rudy. Dear Dr. Català, dear Pere, dear family, friends, and colleagues, I will start from the beginning. It is important to have roots, and Pere is proud of being Catalonian, as a cat is proud of its tail. Pere embarked on his academic journey in 2012 and graduated in 2016 from the University of Barcelona, the beautiful city of Gaudi. He then ventured to the Netherlands, where he enrolled in a master's program in biopharmaceutical sciences at the University of Leiden, Rembrandt's hometown. His academic journey led him then to Utrecht, where another famous Catalonian decorating his thesis, Juan Miró, found inspiration. In 2018, he graduated cum laude and applied for a PhD position on corneal regeneration in a collaboration between our lab at the Merlin Institute for Technology-Inspired Regenerative Medicine and the University Eye Clinic in Maastricht, the famous city of André Rieu. From the very beginning, you demonstrated your curiosity, leaving no questions unasked. I am reading from your first note to us following your interview. I have the following questions. What is the exact role of the Department of Ophthalmology in this project? Which culture techniques would you prefer? 2 or 3-dimensional? And how will success be defined? And do you foresee this project going into in vivo experiments? I shared my impression of you with Vanessa the same day. I think that Pere is motivated and intelligent and has experience with a variety of techniques. Doing science seems like his passion, not only his work. I haven't spoken to the other candidates yet, but in my opinion, it will be difficult for them to surpass the impression that he has made on me. To which Vanessa replied, then let's invite Pere. I also appreciated all the questions he's asking, 3 dots. He is certainly clean, exclamation mark. Your journey was also a new adventure for us, as you were the first PhD candidate that Vanessa and I guided together. 2 kapiteins op 1 schip, we say in Dutch, or too many cooks in the kitchen in English. These expressions are metaphorically used to describe such situations. But under the right circumstances, it is remarkable what co-guidance can achieve. We try to give you the benefit of our diverse competencies, backgrounds, and perspectives. Although we recognize how daunting it is that your supervisors can be in 2 places at once, literally, judging by your achievements, you made the most out of it. You quickly acquired the surgical skills to harvest corneal endothelium at the Eurotissue Bank and set up successful endothelial cultures in Maastricht. You made cutting-edge techniques your own, and your work resulted in the first single-cell transcriptomic atlas of the human cornea. Subsequently, you harnessed this platform to explore the heterogeneity of cultured corneal endothelial cells. Collaborating with expert bioinformaticians, you unveiled the corneal endothelial cells' proliferative capacity across different species. Your achievements have been recognized by top-tier journals and culminated in a highly regarded commission review on corneal regeneration for progress in retinal eye research, which is the highest impact factor journal in our field. In 2020, you were awarded the Bio-Ophthalmology Research Award, and your AVO presentation in 2021 received acclaim for key opinion leaders. Currently, our research group has grown considerably, and you have been pivotal in training our next generation of scientists. To answer your job interview question from 2018, Pere, your work forms the basis for an ongoing line that we wish to take to the clinic and treat patients with corneal endothelial disease. Besides your scientific progress, it was a pleasure for us to watch you grow as a person. During the pandemic, you displayed resilience, adapting and persevering to continue your work to the best of your abilities within the constraints of restrictions. With Mariona, you found love and companionship, and even though Vanessa and I tried our hardest, we still can't recall the name of the cat who tipped the balance on your decision to head back to Utrecht. Pere, it was a pleasure to be your supervisor all these years, but I would like to emphasize the role of Vanessa, without whom this journey would never have been possible. Although we were your daily supervisors, Rudy's door was always open to you, and you returned from your talks full of motivation. All three of us wish you success in your future career, being it in academia, industry, or anything else you consider for yourself. I'm very happy that I'm given the opportunity to be the first person to congratulate you with obtaining your PhD title. Felicidades, Pere, on these achievements and the bright future which lies ahead.
Thank you, Dr. Dickman, for this nice laudation. And coming back to the candidate who's now Doctor, dear Dr. Català, also on behalf of the University of Maastricht and all the deans, I congratulate you with the degree you have acquired. I hope you can enjoy it. Today, a great party. I wish you all the goods for the future and a great academic career, of course. I extend my congratulations to your team, Professor Nuits, Dr. Lapointe, Dr. Dickman, and of course to family and friends here present and online. I would also like to thank the members of the opposition for all their work, and of course the technical support team and the PEDEL who made this hybrid session possible. So before I close this meeting, I have a few announcements. We are going to ask you when I close this session that you go to the refter where the reception is, then you get something to drink. We stay here with 1 or 2 photographers because we want to take a picture with the 2 professors online, and afterwards at the beginning of the steps. So that's what we're going to do while the rest is having a drink. And then we come back to the refter. And if you think, when can we congratulate a candidate? It's quite simple. We go first and you follow us. Okay? Having said that, I close this meeting.